could you give us an idea of the kind of research that you do, what your lab has been focused on, and then yeah, we can kind of just start there.
Sure. So historically, I would say that I was very much interested in how the brain controls eye movements, right? Like 5% of the people in the world have abnormal eye movements. My eyes are not aligned. They don't align together. They don't move together. So it's called strabismus, as a technical term. And so I was always interested in that, and I wanted to know why that was the case. So for the longest time, I thought it was mechanical, like the muscles that move the eyes were, had some abnormal processing going on. But then when I was in undergrad, I did some reading up on it, and I soon learned that it was really not in the muscles, but it was in the signals that come from the brain to the muscles. And so for my PhD, I joined a lab that was focused on motor control and eye movements in particular. And so that was sort of how I got started. But then as over the last 2 decades, as I've done more and more research, I became more interested in the language that the brain speaks specifically for motor control or for movement, and less so focused on eye movements per se. So now, these days, I would say my interest is really to understand how the brain speaks. What is the language of the brain? So let me give you an example that sort of really, I think, hits the point. So let's say I say something to you in Hindi, "Tamaru naam sunche," and then I say the same thing to you in English, "What's your name?" Right? So let's assume that whatever it is that I delivered, I'm a group of neurons, and I represented and I transmitted a message, and then I transmitted the same message again, but in a different language. So I was able to represent different dimensions of information, but you as a decoder, suppose that we are anatomically connected and I'm sending you these signals, you are able to decode only one and not the other. Mm-hmm. So that means I'm multilingual and you are able to only speak a subset of that language, right? So right. I'm interested in knowing how is it that I can represent multiple dimensions of information, or how I can speak in different languages, and what is it that allows you to decode only a certain aspect of it. Now, I do that in the context of sensory motor transformations, and I'll tell you what I mean by that, but it's the same idea. Neurons in the brain, when you orient to your visual environment, there are so many neurons in the brain that respond when a stimulus is presented in the receptive field, right? So there is representation, sensory information encoded in them. So if a target is presented where I like it, this neuron will fire and that's a stimulus response, a sensory response. But these same neurons also fire when you make an eye movement to that location, or when you reach to that location. So these are called visual motor neurons or sensory motor neurons. They can encode both sensory information and motor information. Neurons like this live in the colliculus, the superiorcolliculus, where I do most of my studying, my research. So, and these neurons project to the brainstem, to the brainstem neurons that tug on the muscles, right, if you will. So
all
when
kind
the
of
sensory,
like efferent mediated stuff.
Yeah, so when I put out sensory information and you're the brainstem neuron, you don't move the eyes. But when I put out motor information, you move the eyes, right? So how is it that the decoder can differentiate between sensation and action-related activity? Those are the kinds of questions that I ask now.
Okay.
And
awesome.
Yeah, and in a follow-up paper that's on bioRxiv, I think I sent you that one as well. We actually used your technology to sort of differentiate, to deliver pulses along these electrodes according to what we think is sensory code and what we think is the motor code. And so we were able to use the ability to deliver individual pulses on each contact through your software, and then get at differentiating between sensation and action.
Awesome. Yeah, I want to get into some nitty gritty of kind of the technical usage that you set up for a couple of these experiments, because I think it's a lot of them seem pretty novel again. But I'm interested at a higher level, what some of your work you think could, what it could affect clinically. Obviously, you mentioned strabismus. Yes, I can't believe I forgot it because I actually had strabismus surgery as a child. So yeah, definitely strabismus is probably one of those. What other kind of clinical areas do you see this work potentially affecting? I think I saw you said in one of your papers, just understanding the neural basis for this can help improve neural control for artificial limbs. And then you also mentioned a little bit about ADHD and schizophrenia.
Absolutely, and even Parkinson's. So the research I do nowadays, I wouldn't associate it much with strabismus, but the research I do now is much more relevant to clinical disorders, neuropsychiatric disorders, and motor control disorders. So, for example, one of the things that I believe is going on is that when you have a population of neurons, the relative temporal structure in that population is super important for it to be functionally meaningful. And in motor control, for example, if that population of neurons has temporal coherence, right? So they're working in unison, let's put it that way in simple terms, then it's a potent signal for motor generation. And so what I think happens, for example, in Parkinson's is that this population structure is altered or has gone awry. So there are times when you don't get temporal structure when you need it, but then when you have tremor, it might be that these neurons go into these temporal bursts and as producing these tremor kind of signals. With respect to neuropsychiatric disorders, ADHD is a big one that's been linked with eye movement work, and that is one associated with impulse control, right? That's ADHD. You don't have this ability to inhibit movement. And so what I think happens, or what we've proposed in some of our papers, is that when the signal in the colliculus, for example, with respect to eye movements, goes into this high temporal structure burst mode too early. And so you ramp up and you go into this burst mode and you make a movement, even though when you're not supposed to. So you lack the inhibition to clamp down on the circuit that takes you into this high burst mode. So that's what I think. Those are the angles that we use and motivate for the clinical aspect of our work.
Gotcha. Yeah, I know one of your papers talked about, it had this analogy of like brakes and gas in a car, which was the exact same analogy my PI used for me in kind of autonomic control with like sympathetic and parasympathetic mediated, actions. And so I like that analogy has kind of been used consistently across different areas of neuro research. I've seen that a lot of your work talks about these saccades. I don't know if I'm pronouncing that right, but they are, as you put it, kind of these ballistic eye movements. You're going from a relative stationary point to a very fast movement otherwise. Could you give us a little idea of what some of the typical eye movements in your area of research are, and then kind of why saccades is so relevant to the research you've been working on?
Sure. So saccades, some people call them saccades, potato kind of thing. So either one works. Saccades are, or I call them saccades. So saccades are this ballistic eye movement that's the oculomotor equivalent of reach, right? So it's like point-to-point movements. They're sort of the easiest to study. They're also the most common type of eye movement. So if you're looking around in your visual world, as you're looking on your screen while we're having this conversation, your eyes ordinarily would move around and you would make 3 saccades per second. Sometimes, not sometimes, even when you're fixating, you're making these small movements called microsaccades that are happening. And those two are happening on the order of about 3 times a second. So they're the most prevalent type of eye movement that's out there. And that's one of the reasons that we study it. The other types of eye movements that you may have heard of is called smooth pursuit. So if an object is moving you want to track it. So that depends on a very different mechanism, one in which you have to sort of integrate the speed and the velocity of the target so that you can track it. Then there are other movements called vergence. So in saccades, the two eyes move together. In vergence, the two eyes move towards each other or away from each other. So it's as if like you're looking at something far away and then you look at something closer, or vice versa. And then there are a couple of other types of eye movements as well that are quite common. And by and large, there is a lot of segregation in the neural circuits that are associated for these types of eye movements. But as you go closer and closer and deeper in the brain, there's also a lot of overlap. I think saccades are easier to study because they're ballistic. and they, you want to take the velocity component out, and when the eyes move slow, vision comes into play and things of that sort. So even though I'm calling them ballistic, they're actually not ballistic. You can stop a saccade in mid-flight and then release the gate, if you will, and then the saccade picks up and goes where it needs to go. So there is this whole body of literature that says that there is internal feedback, like used even within to control the accuracy of saccades.
Awesome. Yeah, and by gate, you kind of mean like these inhibitory mechanisms that we have?
Yep, that's exactly right. So brake and pedal, right? You think of CPG, you kind of oscillate between two functions, let's say left and right. Same thing with saccades. When you're looking in your visual environment and you're scanning your world, you're making saccades and you're fixating. You're scanning, you're making a saccade and you're fixating. So fixation is like the brakes, like stop your eyes, and saccades are like moving your eyes. And so these two are just working as a alternating pattern of excitation and inhibition. Yep.
I want to talk a little bit more about kind of the tasks that you've set up for some of these, because I think they're very unique compared to kind of normal NHP tasks. And as I've worked with more NHP research, it seems like their task is really kind of the fundamental linchpin sometime in their experiment. You have to train these animals up and make sure that the task is in fact isolating the thing you want to look at. One of your papers kind of went through a fixation perturbation, I believe is what you called it. And you kind of delivered an air puff to the animal's eye to kind of reset, I believe it was the neural correlates for that ballistic firing. And you kind of called it the blink reflex. Can you kind of tell us about why you made this experiment set up like that, and then also kind of give us an idea of maybe some out of the box NHP tasks that you've seen previously in some areas?
Sure. No, it's an awesome question, and it's one of these things that had been in the back of my mind for a couple of decades until I came across a solution. So think of it this way. Before we learned about the temporal structure argument that I just told you about, we were basically thinking that there's a gate to the saccadic system, right? And this gate needs to be released in order to make an eye movement. What is the earliest I can get an animal to make an eye movement? That's how we were thinking about this. So I've always wanted to remove this gate with temporal precision, right? So I can't go in there and inject lidocaine on it, because lidocaine knocks out the area for 45 minutes. I can't put mucimol in there because it takes it out for hours. Optogenetics is fantastic, except it doesn't really work in monkeys, right? So I wanted some way to be able to remove this gate for a short amount of time without, and reversibly, right, with good temporal control. So it turns out that when you blink, what happens is the eyes move a little bit underneath the eyelid, and this gate turns off. So this has been reported. There's a paper out there that says the gate, that these neurons that are the gates turn off when you blink. So I said,
Wow,
allow
that's
me,
super helpful for
right,
what you're trying to
exactly.
ask.
So there's a low tech way of doing this, and that is deliver a puff of air to the eye at a random time, make the animal blink. And while the animal is doing some task, you can see now, when can you get an eye movement prematurely? It's sort of like, I would say, if I could do this in Vegas, I'd be super rich, right? If I can get the dealer to show me their cards before they should, I could be a billionaire. And so that's sort of how I thought about doing this with monkeys, just sort of get them to see if I can do this experiment to see if I can get them to prematurely reveal what they're planning. And then we saw this beautiful time course of when actually the animal is planning the movement. And so the idea was to perturb fixation and see what happens.
That's awesome. So you had mentioned earlier that the superior colliculus is kind of like your go-to area for the research that you do. Can you tell us a little bit about the SC, where it is in the brain, and then I guess why you ended up going with some of the electrodes that you do? I know that you have some LMA from alpha omega probes and a Plexon V probe. What was kind of the reasoning in that probe selection, and how does it help you in your research?
Yeah, that's a loaded question. I could probably talk about it for an hour if you wanted me to. So phylogenetically speaking, right, evolutionarily speaking, the colliculus was the mecca of the brain before there was cortex. So there's a lot of things that the colliculus does. So if you look at rodents, for example, or even frogs and things like that, the colliculus is at the top of the brain and sort of at the top of the hierarchy in terms of many things that they do. It was the premier site, and it still is, for interacting with the environment, right? Taking in sensory information and acting on it. So the colliculus is also known as the tectum. It's a big player in fish, even the people who do larval zebrafish work and things like that with all these cooler technologies. They work on the tectum for sensory motor integration. So it's a preserved structure that does amazing things. Now, it's a laminar structure. So you just imagine like you've got a bunch of sheets put stacked up. There are 7 sheets stacked up. And the idea is that there are sheets. So in the visual cortex, if in the monkey, for example, that's on the surface here, but it's within the sulci. And the cortex has lots of folds and gyri and all of that stuff. So it's not laid out cleanly, whereas the colliculus is laid out cleanly. The visual cortex has a map of the visual world. So does the colliculus.
If you could design a new kind of experiment, so you've played around with adding additional breaks, you've played around now with kind of taking these breaks all the way away. Is there kind of a next experiment that you would love to be able to test a new part of that system?
Yeah. So this temporal structure aspect is something that I'm high on right now. That's sort of at the forefront of my mind. And so what I'd like to do is train the animal to volitionally change the activity of their neurons. My notion is that if I can get the animal to rev up the activity of these neurons together, the animal will make a saccade, even though he's not supposed to, even though I've never told him to.
What got you into this field, starting like high school, undergrad? What made you interested?
So I sort of alluded to that already. I remember I must have been, I don't know, 7, 8, maybe 10 or 12 years old. I was laying in bed at my cousin's house, and he had a poster up of like NASA astronaut or something like that. And I blurted out that I'm going to be a scientist someday. And it amazes me that I remember that and it's come true. So that was sort of probably my first thought that there was something in this direction for me. as I mentioned earlier, my own issues with strabismus is what paved the path for me, right? I was always interested in that. I thought so I started in biomedical engineering as an undergrad at Boston University, just like Lee. And I was interested in biomechanics for reasons I mentioned, and I think through some readings and things like that, I learned that it was not the muscle, it was the brain. And when I went to grad school, I got the opportunity to work with a fantastic mentor, Edward Keller, who's now retired. He trained with someone who's considered the Pope of oculomotor work, David A. Robinson, back in the 60s. They trained, and so I had the perfect mentor to coach me through this, and that's how I got into it. That set up that set the path for me.
What you do outside of work?
I work a shitload. In addition to being running a lab, a monkey lab, I teach, which is all necessary, but I'm also the graduate program director. So I am in charge of the entire PhD program once the students come into the program. And I also run a new master's in neural engineering program that I developed. So I basically wear 3 hats at work, and that keeps me busy. So when I'm not doing that, I will spend time with my family, and if I'm enjoying a good drink, I love single malts. Single malts is, I could just enjoy them forever. My other goofy hobby is I love playing killer Sudoku. Not just regular Sudoku, but it's got to be killer Sudoku. And online, there is a website, there's a puzzle of the day every day. I have to make sure that I get to it. And I see you guys checking it out right now. Killer Sudoku online. I have to do puzzle. I haven't done it today yet. I'm itching.
How is killer Sudoku different from normal Sudoku?
So at the end of the day, it's exactly the same rules, right? 1 through 9 in each column and each row and in each grid. But what they tell you is they have a dotted line around a few boxes, and they tell you the numbers that go in this dotted boxes add up to a certain number. So that's the constraint.
Oh, okay.
Yeah, and it makes it, and there are different levels, obviously, and there are times when I can be done in 10 minutes, and there are times when I can spend an hour and not get it done. So
it's more about time.
Yeah, Sunday is the hardest.
Well, hopefully you have the most time to work on it on a Sunday. I don't know how you prioritize those 3 jobs you're working. So
yeah, exactly. So Sunday morning, I'm a morning person, so I'll get up in the morning, I'll sit in front of my computer, do some reading and, see what's going on in the world, play my Sudoku, and then go through my emails and stuff like that. So, yeah. But I've got a problem, the addiction to killer Sudoku. I almost agreed to admit to it.